When Africans talk about theft, the world expects us to point to obvious things: gold, cobalt, oil, bronze, diamonds, land. But what happens when the new resource is invisible? What happens when extraction no longer arrives with rifles and flags, but with PPE suits, chain of custody forms, dry ice, and international courier labels? What happens when the thing leaving Africa is not ore from the ground, but blood from the body? That is the uncomfortable question of this video. During and after the West African Ebola crisis, hundreds of thousands of biological samples were collected. Investigative reporting by Emmanuel Freudenthal estimated that over 269,000 diagnostic blood samples were taken during the outbreak, and that many were sent abroad for research, while affected countries and many local scientists later struggled to determine where those samples had gone and under what rules they were being used. So this is not just a medical story, it is a story about sovereignty, a story about scientific ownership, a story about who controls biological data once it leaves the communities from which it came. And above all, it is a story about the modernization of colonial logic, because the old scramble for Africa did not end. It learned how to use a centrifuge. If you value stories that go deeper than headlines, subscribe. On this channel, we look at the systems beneath the story. Now let's open the file. Part 1. From Looted Bronze to Looted Biology. In 1897, British forces looted Benin's bronzes and carried them into imperial museums and private collections across Europe. The theft was physical, obvious, undeniable. The objects left the kingdom, entered another legal system, and were transformed from sacred and political heritage into collectible art. That pattern should sound familiar, because the deepest structure of colonial extraction was always the same. Take something of value from Africa, move it into an external system, reinterpret it using foreign law and foreign institutions, and then make Africans negotiate to access what was once theirs. Today, the object has changed. The mechanism has not. Now, the thing extracted may be a pathogen sample, a tissue specimen, a genome sequence, or the digital code derived from that biological material. The international legal debate around digital sequence information, or DSI, exists precisely because genetic resources can now be digitized, shared globally, and used in research and product development far from where the original material was collected. The Convention on Biological Diversity and the Nagoya Protocol were created to promote fair and equitable benefit sharing from genetic resources. And the DSI debate exists because those older rules were strained by modern sequencing and data sharing technologies. This matters because once a pathogen or blood sample is transformed into sequence data, the original African body disappears from the legal imagination. The patient becomes a data point, the community becomes a source site, the outbreak becomes a research opportunity, and the biological history of a place can be converted into scientific property elsewhere. That is why critics call it biopiracy. Not because all research is theft, but because a familiar pattern appears when the source communities carry the risk, the suffering, and the biological burden, while institutions elsewhere capture the knowledge, the publications, the patents, and the long-term strategic advantage. Part 2. Why African biology matters so much. To understand why this happened in West Africa, you have to understand why African biological material is so valuable in the first place. Africa is central to the story of human genetics. The Human Heredity and Health in Africa Initiative, H3 Africa, explicitly notes that the continent's extraordinary genetic diversity creates enormous opportunity for genomic research relevant both to African populations and to global health more broadly. H3. Africa was built in part because African scientists and institutions understood that if this diversity was not studied with African leadership and infrastructure, it would continue to be mined by others. That diversity matters in at least 3 ways. First, it matters for basic science. If modern humans originated in Africa, then African populations contain some of the deepest and richest reservoirs of human genetic variation. Second, it matters for infectious disease. Pathogens do not exist in a vacuum. They interact with hosts immune systems, environments, and long evolutionary histories. Third, it matters for product development. Precision medicine, vaccine design, diagnostics, and genomic surveillance all become stronger when they include diverse populations instead of building from a narrow genetic base. In other words, African biology is not marginal to the future of medicine. It is central to it. And whenever something is central to the future of medicine, it is also central to profit and power. That is where the humanitarian story begins to split. Because one side says, we need samples to save lives. The other side asks, who owns what those samples become? Both questions are real, and the collision between them is where the politics begins. Part 3. The Ebola Crisis and the Great Sample Extraction. During the 2014 to 2016 Ebola epidemic, West Africa became the site of the largest Ebola outbreak in recorded history. The national academies recorded more than 28,000 cases and over 11,000 deaths, and emphasized that the outbreak exposed major weaknesses in global infectious disease preparedness. In that context, sample collection expanded rapidly. Blood had to be drawn for diagnostics. Surveillance systems had to be built. Testing laboratories had to process huge numbers of suspected cases. That much is straightforward. But the scale matters. Freudenthal's investigation estimated that over 269,000 diagnostic samples were collected from patients across Guinea, Liberia, and Sierra Leone. He also reported that many people were never properly informed that their samples could later be used for research, and that samples were sent to laboratories across the United States, the United Kingdom, France, Germany, Canada, Australia, Hungary, and elsewhere. Wired later amplified the story, summarizing the Telegraph's reporting and noting that many of the samples that the public assumed had been destroyed were instead believed to remain in the custody of government agencies and possibly pharmaceutical companies and labs in Europe and the United States. The concern was not simply that samples moved, it was that the countries from which they came might have little say over downstream research or any commercial products developed from them. This is the moment where a humanitarian emergency becomes something else. In the middle of crisis, consent becomes blurry, documentation becomes rushed, rules become flexible, samples move under emergency logic. Then after the cameras leave, those samples do not necessarily come home. They enter research pipelines, repositories, databases. And with that, the moral problem changes. Because emergency collection may be defensible. Indefinite external control is a different matter. Part 4. The Fort Detrick Connection. Now let's talk about one of the names that turns this from a public health story into a geopolitical one. Fort Detrick. Fort Detrick in Maryland is home to the U.S. Army Medical Research Institute of Infectious Diseases, or USAMRIID, and to high containment research facilities that have long been central to the U.S. biodefense system. USAMRIID's own public materials describe capabilities for analyzing clinical, environmental, and biological material for dangerous pathogens, including sequencing and other advanced molecular procedures. This matters because USAMRIID and Fort Detrick linked institutions were not on the sidelines during Ebola. Army and Defense Department materials show that USAMRIID personnel deployed to Liberia and Sierra Leone during the outbreak helped establish testing capacity, train local personnel, supported diagnostics, and later set up genomic surveillance systems in collaboration with the Liberian Institute for Biomedical Research. The U.S. Army also publicly stated that efforts to sequence Ebola genomes had previously been slowed by the need to send samples back to the United States, which is one reason field sequencing capacity was considered so important. Peer-reviewed papers connected to the response make this even more concrete. A Journal of Infectious Diseases paper on Ebola diagnostic testing in Liberia states that 547 human clinical blood samples and oral swabs collected in Liberia between November 2014 and January 2015 were tested by teams from the Liberian Institute for Biomedical Research, the U.S. Army Medical Research Institute of Infectious Diseases, and the NIH Integrated Research Facility. So we need to be precise. The documented record does not support saying that all or even most, of the 269,000 plus samples ended up at Fort Detrick. But it absolutely does support saying that Fort Detrick-linked U.S. biodefense institutions were directly involved in outbreak diagnostics, sequencing, and analysis tied to West African Ebola response. That changes the political meaning of the story, because once military-linked biodefense institutions become part of the sample and sequence chain, the issue is no longer only medicine. It is also national security. To a biodefense system, an Ebola sample is not just a tragedy from someone's bloodstream. It is a dual-use asset. It helps answer questions like, how does this pathogen evolve? How quickly is it mutating? Will existing diagnostics still work? Could therapeutic targets fail? What genomic signatures matter in future outbreak response? Those are valid scientific and defensive questions, but they are also strategic questions. And when the strategic value is extracted from African crises, while the source countries lose control of their own biological material, the sovereignty question becomes impossible to ignore. Part 5. From sample to sequence to property. And this is the point in the story where many viewers instinctively ask, how do you turn blood into power? The answer is surprisingly simple. First, you collect a sample. Then you isolate or characterize the pathogen. Then you sequence the relevant genetic material. Then that sequence can be stored, compared, modeled, and used to build diagnostics, therapeutic approaches, vaccine candidates, surveillance tools, and future patents. At that stage, the resource is no longer the vial itself. It is the information. This is why the DSI debate is so important. The Nagoya Protocol was built around fair and equitable benefit sharing from genetic resources, but biotechnology made it possible to extract the useful value of a sample into data that can travel globally without the original material. That is exactly why debates over digital sequence information have become so intense. If the sequence can be shared or commercialized without meaningful obligations to the source country, then the old promise of equitable benefit sharing becomes hollow. This is where the language of a genetic goldmine becomes useful, because what is being mined is not just the liquid in the tube, it is the code hidden within it, and code is more scalable than a mineral. You can copy it infinitely, store it remotely, commercialize it across jurisdictions, integrate it into product pipelines, and in many cases, the source community never regains control. That is what makes biological extraction in the digital age more dangerous than old forms of looting. The bronze leaves once, the sequence can circulate forever. Part 6. The pharmaceutical profit side. Now let's follow the money. There is a common myth that public health emergencies and pharmaceutical profit occupy separate moral universes. They do not. They overlap constantly. Ebola provides a powerful example. The first licensed Ebola vaccine, now known as Ervibo, originated in public sector science. It was initially engineered by scientists at Canada's National Microbiology Laboratory, then licensed to a U.S. biotech company, and later exclusively licensed onward to Merck in late 2014, at the height of the West African outbreak. Merck's own materials say it obtained exclusive worldwide rights to research, develop, manufacture, and distribute the vaccine candidate. A legal history analysis of the vaccine's development notes that relevant patent applications were granted in Europe, the United States, and Canada between 2010 and 2013, and that the project later moved through licensing agreements, U.S. government support, and corporate development pathways. In other words, the public health emergency was also a moment in which intellectual property, licensing rights, and future market control solidified around a product platform. Now, to be fair, Merck and public agencies did help push a vaccine across the finish line. That matters. But the structure also matters. A crisis in Africa accelerated the urgency and global value of Ebola countermeasures. Clinical trials and field use took place in African populations, including large ring vaccination efforts in Guinea and later deployment in the Democratic Republic of Congo. So the profit side is not a cartoon where one company simply steals blood and prints money. It is more sophisticated. It is the conversion of crisis into product pipelines, the conversion of public danger into patentable solutions, The conversion of outbreak urgency into licensing value. The conversion of African biological reality into commercial leverage. That is the pharmaceutical loop, and Africa often enters it at the raw end, as sample provider, as trial site, as emergency theater, not as equal owner of the downstream intellectual property. This is exactly what critics mean when they say the system treats African biology as global commons at the point of extraction and private property at the point of profit. Part 7. The ethics of consent and the problem of emergency. At this point, defenders of the system usually make the strongest counter-argument. They say, in an outbreak, you cannot run perfect consent procedures. You cannot halt urgent sample transfers. You cannot build international response on lengthy legal negotiations. And again, there is truth here. Outbreak response is messy. Lives are on the line. Diagnostics cannot wait. But the fact that emergency complicates ethics does not erase ethics. It intensifies them. That is why reporting on Ebola sample transfers caused such controversy. Multiple investigations and commentaries argued that patients and families often did not understand that their samples might be used long after diagnosis, and that affected governments themselves lacked clear information about where materials had gone and what future research they would support. This is what some writers and African commentators call medical colonialism, not because medicine itself is colonial, but because the structure is familiar. The emergency happens here, the extraction happens here, the high-value institutional control happens elsewhere. And once that pattern repeats often enough, trust begins to collapse. That is not a moral side issue. It is a strategic problem. Because outbreak response depends on trust. Communities must believe that science is not simply another route through which their vulnerability will be converted into someone else's advantage. If they stop believing that, they stop cooperating. And once that happens, the entire global health system becomes weaker. So even for people who care only about pragmatic disease control, exploitative sample governance is self-defeating. Part 8. Parachute Science and the Blackout of African Expertise. Now let's talk about the people who are too often erased from this story, African scientists. One of the ugliest features of extractive science is not only the movement of samples, it is the movement of credit. Researchers from wealthy institutions often drop in during emergencies, gain access to precious biological material and outbreak data, then return to publish high-impact papers, build careers, and secure grants. This pattern has become widely known as parachute science, and it is one of the reasons African researchers have pushed so hard for stronger local infrastructure, biorepositories, and data governance. H3 Africa, for example, was explicitly designed to create a sustainable African research enterprise led by African scientists with its own infrastructure, resources, training, and ethical guidelines. By 2024, the consortium included hundreds of researchers across 30 African countries and had built biorepositories in Nigeria, Uganda, and South Africa. That is the resistance side of this story. Because African scientists are not merely complaining, they are building alternatives. They are saying correctly, if Africa's genetic and pathogen data matter so much, then African institutions must not remain glorified field collection zones for Northern science. They must be labs, repositories, data centers, publication hubs, regulatory actors, owners. That is what scientific sovereignty means. Not isolation, not anti-collaboration. Ownership within collaboration. Leadership rather than permanent extraction. Part 9. Fort Detrick, Biodefense, and the Sovereignty Question. Now let's return to Fort Detrick one more time, because it sharpens the stakes. The U.S. Army's own materials make clear that USAMRIID sees dangerous pathogens through a biodefense lens and works on diagnostics, sequencing, and analysis relevant to biological threats. This is not scandalous by itself. Every serious state has a public health security interest in understanding dangerous pathogens. The real issue is different. What happens when a foreign, military-linked biodefense ecosystem accumulates detailed biological data from African outbreaks faster than African states can build their own equivalent systems? What happens when the strategic knowledge generated from African crises is stored, interpreted, and weaponized, even defensively, outside African institutional control? That is the sovereignty nightmare. Because at that point, African biology is no longer only part of a medical story, it becomes part of another nation's strategic archive, a reference library for defense planning, a repository of outbreak behavior, genetic variability, diagnostic targets, and future countermeasure design. And once that information asymmetry becomes deep enough, Africa's dependency grows. The continent becomes essential to the world's understanding of certain pathogens, yet less able to command the terms on which that understanding is built. That is not only unfair, it is geopolitically dangerous. Part 10. The New Battle, Pathogen Sharing, Data Sharing, and Africa, CDC. This debate is not stuck in the past. It is happening right now. In 2026, Reuters reported that the head of Africa, CDC, raised major concerns about U.S. health agreements with African countries because of clauses around pathogen sharing and data sharing, warning that some proposals required countries to promptly share pathogen data with Washington without clear guarantees that resulting vaccines or drugs would return on equitable terms. Zimbabwe reportedly withdrew from one such negotiation, and Zambia also pushed back on certain clauses. That is an extraordinary development because it proves this is not just an old Ebola-era ethics dispute. It is a live geopolitical struggle over the terms of biological cooperation. Africa CDC has been building its Institute of Pathogen Genomics and the Africa Pathogen Genomics Initiative precisely because the continent understands that sequencing capacity and data infrastructure are not luxuries. They are pillars of sovereignty. Africa CDC explicitly frames pathogen genomics as essential not only for surveillance and outbreak response, but also for vaccine, diagnostic, and therapeutic development, and warns that Africa still lacks enough infrastructure, tools, and governance frameworks to fully capture those benefits on its own terms. That is the future battle line. Not whether samples will ever cross borders again. They will, but under whose rules? With what consent? With what benefits sharing? With what authorship rights? With what return of value? If Africa does not answer those questions now, others will answer them for it. Part 11. Is biological data a national resource? We have reached the point where we need to ask the big question directly. Should DNA, pathogen samples, and sequence data be treated as protected national resources in the same way countries treat oil, gold, or strategic minerals? There are good reasons to hesitate. Pathogens do not obey borders. Global science benefits from speed. Open sharing can save lives. Those are real concerns. But there are also good reasons to say yes, because biological data is not neutral once it enters systems of patents, product development, military biodefense planning, and unequal bargaining power. At that point, it becomes strategic. And if it is strategic, then sovereignty matters. The African answer does not need to be isolationism. It needs to be structured cooperation. Samples can be shared, data can be shared, but the terms must change. That means prior informed consent, clear governance, traceability, and those systems increasingly define the future of medicine, security, and wealth. So if Africa does not guard its biological borders, the next century of extraction will not be about what is under the ground. It will be about what is inside us. If this changed how you think about science, sovereignty, and the new scramble for